Okay, let's start our today's topic. So our today's topic is enzyme inhibition. We were discussing it yesterday. Yesterday we discussed that enzyme inhibition is of two types. One is competitive inhibition and another is non-competitive inhibition. In competitive inhibition, a substrate molecule and an inhibitor molecule competes for occupying the active site of enzyme. Then I cited you an example of this competitive inhibition. The most famous example of competitive inhibition is this competition between para-aminobenzoic acid and sulfur drugs, sulfonamides, for occupying the active site of enzyme, which is involved in the synthesis of folic acid. Now, today we are going to discuss about non-competitive inhibition. As I told you yesterday, this non-competitive inhibition, it is of two types: irreversible non-competitive inhibition and reversible non-competitive inhibition. So, first we will discuss irreversible non-competitive inhibition. Irreversible. Irreversible non-competitive inhibition. To explain this, I would like first to tell you about how our muscles contract. You know, this is a muscle cell. This is the plasma membrane of a muscle cell. The plasma membrane of a muscle cell is known as sarcolemma. Sarcolemma. It is sarcolemma of a muscle cell. These muscle cells, they contract only when they receive nerve impulses from our nervous system. So a nerve fiber brings a nerve impulse towards this muscle cell. This is a nerve terminal. This is a nerve terminal. End of a nerve fiber. Nerve terminal. You know, these nerve fibers, they do not actually touch our muscle cells. There is a gap between the end of this nerve fiber and the muscle cell. This gap is known as synaptic cleft. Synaptic cleft. So how do muscle cells contract? So when nerve impulses reach to muscle cells, these nerve impulses make the muscle cells to contract. Since the nerve terminal and the muscle cell, they are not in touch. In between them, there is a gap. That gap is known as synaptic cleft. Then how this nerve impulse is transmitted to a muscle cell? So for that purpose, this nerve terminal, it has got a knob-like swelling. This is known as synaptic knob. Synaptic knob. In this synaptic knob, this nerve terminal has got some vesicles, fluid-filled vesicles. These fluid-filled vesicles, they are filled with a fluid. These fluid-filled vesicles, they are known as synaptic vesicles. Synaptic vesicles. In these synaptic vesicles, there is a substance known as a neurotransmitter. There are chemicals known as neurotransmitter. So when a nerve impulse arises, when a nerve impulse reaches, to this synaptic knob, this wave of depolarization, how we define a nerve impulse? We define a nerve impulse as a wave of depolarization traveling along the length of a nerve fiber, right? So this wave of depolarization, when it reaches to synaptic knob, it stimulates these synaptic vesicles for exocytosis. So these synaptic vesicles, they join with the neurulema, covering membrane of the nerve fiber, and they release some chemicals in this gap. So which chemicals they release in the gap? They release the chemicals which were stored in them. Which chemicals were stored in them? So they release neurotransmitters. They release neurotransmitters in this gap. As a neurotransmitter is released, it causes depolarization of the sarcolemma. So far, the sarcolemma was polarized, it was charged, right? Now, as the neurotransmitter is released, it causes depolarization of sarcolemma. So, sarcolemma gets depolarized. This sarcolemma gets depolarized. This wave of depolarization, it goes deep into the muscle cell. It reaches to SR. Wave of depolarization reaches to SR, sarcoplasmic reticulum. And sarcoplasmic reticulum causes release of calcium. Sarcoplasmic reticulum releases calcium. This calcium, it binds to a protein in the sarcoplasm of this muscle cell. That protein is known as troponin protein. It binds to troponin protein. And this binding of calcium to troponin protein, it causes the muscle contraction. It stimulates the myofilaments. It stimulates the myofilaments for contraction. For contraction. And the muscle contracts. And the muscle cell contracts. So this is how our muscle cells contract. So for muscle contraction, we need neuroimpulses from our neuroimpulses, from our nervous system. So neuroimpulses, they are generated by the nervous system, and this nervous system, it sends these neuroimpulses to muscle cells. To muscle cells. This is a muscle cell. Through its branches known as neurofibers or neuroterminals. So when a neuroimpulse reaches to a muscle cell, it reaches in the form of a wave of depolarization. So when this wave of depolarization reaches to the end of this neurofiber, reaches into the synaptic knob, it stimulates the synaptic vesicles. Neuroimpulse stimulates synaptic vesicles to go for exocytosis. To go for exocytosis. Synaptic vesicles, they go for exocytosis. They release neurotransmitters in this synaptic cleft. A release of neurotransmitters in the synaptic cleft causes depolarization of the sarcolemma. Sarcolemma gets depolarized. When sarcolemma gets depolarized, this wave of depolarization travels into sarcoplasmic reticulum of the muscle cell. Sarcoplasmic reticulum releases calcium. Calcium binds to a protein on myofilaments known as troponin protein, and this stimulates the myofilaments for contraction. When the myofilaments contract, the whole muscle cell contracts. When one muscle cell contracts, another muscle cell contracts, the whole muscle contracts, right? So this is how a muscle contracts. Then how do these muscles relax? These muscles can relax only if the neurotransmitter is removed from this synaptic cleft. Since it was the release of neurotransmitter which depolarizes the muscle cell, which causes the contraction of the muscle cell. Now, if the muscle has to relax, this neurotransmitter, it must be removed from this synaptic cleft. So how this neurotransmitter is removed from the synaptic cleft? It is removed from the synaptic cleft by the help of an enzyme. So that enzyme in this case is acetylcholine esterase. This neurotransmitter in this example, the neurotransmitter which is released here in this example is acetylcholine. Acetylcholine. Now, if we want to remove this neurotransmitter, if we want muscle to relax, then this neurotransmitter must be removed from the synaptic cleft. So how this neurotransmitter, acetylcholine, will be removed from the synaptic cleft? So it will be removed from the synaptic cleft by the help of an enzyme. There is an enzyme known as acetylcholine esterase. There is an enzyme known as acetylcholine esterase. What is its function? It converts acetylcholine. It breaks acetylcholine. Acetylcholine esterase converts acetylcholine into acetic acid and choline. So when this neurotransmitter, by the help of this enzyme, acetylcholine esterase, is converted into acetic acid and choline, it is reabsorbed by the synaptic knob. It is reabsorbed by the synaptic knob, and this gap gets emptied. So whole of the neurotransmitter acetylcholine, it is converted into acetic acid and choline by acetylcholine esterase enzyme. And this acetic acid and choline, it is reabsorbed by the synaptic knob, this synaptic knob. And it is again utilized in the formation of acetylcholine in the synaptic vesicle. So this gap gets emptied. When this gap gets emptied, when the neurotransmitter is really removed from this gap, when the neurotransmitter is removed from this gap, this sarcolemma, it repolarizes. It repolarizes. Troponin releases the calcium, and calcium is stored back by sarcoplasmic reticulum. So when calcium is withdrawn back into sarcoplasmic reticulum, muscle contraction stops and muscle relaxes, right? So muscle will only relax when the neurotransmitter is removed from the synaptic cleft. If the neurotransmitter remains there, then muscles are not able to relax, right? So here acetylcholine esterase plays an important role in relaxation of the muscles. Contraction was stimulated by acetylcholine neurotransmitter. But if the muscle has to relax, then this acetylcholine must be removed from the synaptic cleft so that muscle can relax, right? Now, there is an inhibitor molecule which causes irreversible inhibition of this enzyme, acetylcholine esterase. There is an inhibitor molecule which causes irreversible inhibition of this acetylcholine esterase. What is that inhibitor molecule? You might have heard about gas, which is used in biological warfare. Gas. Its chemical name is DFP, diisopropyl fluorophosphate. Diisopropyl fluorophosphate, DFP. It is commonly known as gas. It kills a lot of people in biological warfare. And those people who are killed by this gas, they die with outstretched limbs and wide open mouth. Why they do die like this? Why they die like this? Why they die with outstretched limbs and wide open mouth? So let us discuss that. This gas, DFP, diisopropyl fluorophosphate, it binds to acetylcholine esterase irreversibly. This is acetylcholine esterase enzyme. What was its function? Its function was to convert acetylcholine into acetic acid and choline so that the muscles could relax. This DFP, diisopropyl fluorophosphate, it binds only to a part of the active site of this enzyme, acetylcholine esterase enzyme. This is acetylcholine esterase enzyme. It binds to one of amino acids on its active site. On this active site, there is an amino acid serine. This diisopropyl fluorophosphate, it binds only to this serine. When DFP binds to serine, now this acetylcholine esterase, it cannot bind to its substrate. The substrate acetylcholine, it is substrate acetylcholine. This is acetylcholine. It is substrate. It cannot join to this enzyme. So when gas, DFP binds to a single amino acid of the active site of this acetylcholine esterase enzyme, it causes the enzyme inhibition. So when this DFP binds to serine amino acid of the active site of acetylcholine esterase enzyme, now acetylcholine, it is substrate, cannot bind to it, and the enzyme gets inhibited. Enzyme gets inhibited. Sorry. So when enzyme is inhibited, now acetylcholine, it cannot be broken into acetic acid and choline. It cannot be reabsorbed from synaptic cleft. It remains in the synaptic cleft. Acetylcholine remains in the synaptic cleft. And depolarization of the muscle cell continues. Depolarization of the muscle cell continues. So muscles remain in a state of continued contraction. Muscles remain in a state of continued contraction. So what this causes? It causes a person. It causes a person's death. How? You know, our respiratory muscles, external intercostal muscles, intercostal muscles, internal intercostal muscles, phrenic muscles of the diaphragm. They cause inspiration and expiration by their alternate contraction and relaxation. So if we have to inhale the air, we have to contract external intercostal muscles, we have to contract phrenic muscles of the diaphragm. So when external intercostal muscles contract, they lift the thoracic wall upwards and outwards, decreasing the pressure in thoracic cavity. So that makes the atmospheric air to rush into the lungs. When diaphragm contracts, it moves downwards, increasing the volume of thoracic cavity. That makes the atmospheric air to rush into our lungs, right? So when they contract, they pull this thoracic wall outwards and upwards, diaphragm went downwards, so they caused inspiration. Now, if we have to expire the air, if we have to exhale out the air, we need to relax these muscles. These external intercostal muscles and the phrenic muscles of the diaphragm, they must relax so that the volume of thoracic cavity is decreased and pressure is increased on the lungs, so that lungs are compressed and exhalation occurs, right? But when the enzyme acetylcholine esterase gets blocked, acetylcholine, it remains in the synaptic cleft. It doesn't allow these muscles to relax. So a person is only able to inhale, but the person is not able to exhale, and the respiration stops, and the person dies gasping for the air, right? So person dies with outstretched limbs because of continued contraction of the limb muscles. So person dies with outstretched limbs because of continued contraction of the muscles of the limbs. The person dies with mouth wide open because he is only able to inhale the air. He tries to exhale, but the muscles do not relax. So the exhalation is not possible. And the breathing stops and the death occurs, right? So this is how this enzyme inhibition can kill a person. So if the acetylcholine esterase enzyme is blocked, if it is inhibited, it can cause the death of the person. It can cause death of the person very immediately, right? So this property of the chemical diisopropyl fluorophosphate, it was worked out by the scientists and they used it for biological warfare. They prepared neurogas. They formed neurogas, which is used to kill the soldiers in the battlefield with outstretched limbs and wide open mouth, right? So this enzyme gets inhibited, and this inhibition is irreversible. Once the diisopropyl fluorophosphate binds to serine amino acid, it binds to it permanently. So enzyme gets inhibited permanently. So this, we cannot remove this diisopropyl fluorophosphate from the serine amino acid. So that is why we call it as irreversible non-competitive inhibition. Why it is non-competitive inhibition? Because there is no competition between acetylcholine, the substrate, and the inhibitor molecule. Inhibitor molecule just binds to a single amino acid, serine, on the active site, which prevents the binding of the substrate, acetylcholine, to this enzyme, right? So there is no competition for occupying the active site. So that is why we call it as non-competitive inhibition. It is irreversible because once the DFP binds to a serine amino acid, then it cannot be removed, right? So this is irreversible non-competitive inhibition. And its famous example is this neurogas diisopropyl fluorophosphate. So this is the mechanism how this enzyme works, how this inhibition works, right? Then we have got another inhibition that is known as reversible non-competitive inhibition. Now we have got a reversible non-competitive inhibition. A reversible non-competitive inhibition. This inhibition is also known as allosteric inhibition, or it is also known as end product inhibition, or it is also known as feedback inhibition. So it is known by different names. Reversible non-competitive inhibition is also known as allosteric inhibition. It is also known as end product inhibition, or it is also known as feedback inhibition. So there are certain enzymes in our body which, in addition to active site, have another site well away from the active site. This site is known as allosteric site. So this is the active site of enzyme. This is the active site of enzyme. Away from this active site, there is another site where an inhibitor molecule can bind to it. That site is known as allosteric site. So these enzymes are known as allosteric enzymes. These enzymes are known as allosteric enzymes. Why we call them as allosteric enzymes? Allo means different, and steric means shape. So they show different shapes. They exist in different shapes. So when the inhibitor molecule is not bound to it, the shape of active site is in its natural state. The active site is in its natural form. Active site is in its natural form. When the inhibitor molecule is not bound to this enzyme, the shape of active site is in its natural form. However, when the inhibitor molecule binds to this enzyme, when the inhibitor molecule binds to this enzyme, suppose this is the inhibitor molecule. This is the inhibitor molecule. It now binds to this allosteric site. The shape of active site, shape of its active site is changed. Shape of its active site is changed. In natural form, the shape of active site was like this in the absence of inhibitor molecule. Now, as inhibitor molecule is bound to this allosteric site, this binding of inhibitor molecule to allosteric site causes the active site to change its shape. And now the substrate cannot bind to this enzyme because of the change in the shape of its active site. So this inhibitor molecule it causes the inhibition of the enzyme by bringing about change in the shape of its active site. So we call these enzymes as allosteric enzymes. Why we call them as allosteric enzymes? Because they show different shapes. When they show different shapes, when the enzyme is not having inhibitor molecule bound to it, the active site is in its natural shape or natural form. But as the inhibitor molecule binds to allosteric site, although inhibitor molecule binds to allosteric site, but if it affects the enzyme in such a way that the shape of active site gets modified and the substrate cannot bind to it now. So when the active site's shape gets modified and substrate cannot bind to it now, so we say that enzyme gets inhibited, right? So for that reason, we call them as allosteric enzymes. So these enzymes are known as allosteric enzymes because they are capable of changing their shape. When do they change their shape? They change their shape when inhibitor molecule binds to them. Where the shape change occurs, the shape is changed by the active site. So active site assumes a different shape so that substrate molecule does not bind to it and the enzyme gets inhibited. That is why we call them as allosteric enzymes. And this type of inhibition is known as allosteric inhibition. So for that reason, we call it also as allosteric inhibition, right? Now, why we call it as end product inhibition or feedback inhibition? Let us discuss that. These allosteric enzymes, they regulate various metabolic pathways of our body. They are involved in the regulation of various metabolic pathways of our body. For example, in glycolysis, in glycolysis, glucose is converted into glucose 6 phosphate. Glucose 6 phosphate. So this glucose in glycolysis is converted into glucose 6 phosphate in very first step. And this step, this conversion of glucose to glucose 6 phosphate, it is regulated by an enzyme, hexose kinase. It is regulated by an enzyme hexose kinase. This hexose kinase is an allosteric enzyme. So when the end product glucose is converted into glucose 6 phosphate, when this glucose 6 phosphate accumulates in sufficient quantity, then this glucose 6 phosphate it binds to the allosteric site of the enzyme. Suppose this is the enzyme, then allosteric enzyme. Its substrate is glucose, right? It is converted to glucose 6 phosphate. It converts the substrate. When they bind, when the substrate binds to enzyme, when the substrate binds to enzyme, it converts the substrate into glucose 6 phosphate. It converts the enzyme to substrate to glucose 6 phosphate. And the enzyme itself remains unaltered. Right? So this is glucose 6 phosphate. When this glucose 6 phosphate concentration rises sufficiently, then this glucose 6 phosphate it binds to allosteric site of the enzyme. It binds to allosteric site of the enzyme. As it binds to allosteric site of the enzyme, the shape of the active site of the enzyme is modified. It is here glucose 6 phosphate. Glucose 6 phosphate. it is bound to allosteric site. Now the shape of the active site of the enzyme gets modified. And now this substrate molecule cannot join it. So the pathway is stopped. So for that reason, we call it as end product inhibition. Why we call it as end product inhibition? Because the end product of this hexose kinase was glucose 6-phosphate. Hexose kinase, it had to convert glucose into glucose 6-phosphate. So when it formed the end product, glucose 6-phosphate, the end product accumulated in large quantities. Then one of the end products, one of the glucose 6-phosphate molecule, it binds to allosteric site of the enzyme, hexose kinase, right? When it binds to allosteric site, the active site gets modified. Now no more glucose can join to it, and the pathway is stopped. So this end product causes the inhibition of the enzyme. That is why we call it as end product inhibition. That is why we call it as end product inhibition. We also call it as feedback inhibition. Since the quantity of glucose 6-phosphate had risen to suitable, sufficient amounts, now it is no more required. So end product is in sufficient quantity. It is no more required. Then this end product makes the enzyme to stop its catalytic activity till this glucose 6-phosphate will be utilized. And then this glucose 6-phosphate molecule will be removed from the allosteric site, and the enzyme will again start catalysis. That is why we call it as feedback inhibition, end product inhibition, or allosteric inhibition. So that was all about enzymes. I hope you have understood it. This is section for today. Thank you for being with me.